We will be presenting our project optimizing acid pretreatment of cowgrass for ethanol production. As conventional fossil fuels are declining in supply and bioethanol, a renewable fuel, is already used in Brazil to replace gasoline, there is thus growing production of bioethanol around the world. Cowgrass, meanwhile, is widely available and can be used to produce bioethanol. However, pretreatment is needed to remove lignin and hemicellulose and to expose the cellulose for cellulase hydrolysis. Current pretreatment methods include acid pretreatment, alkaline pretreatment, and steam explosion. The pretreatment, however, needs to be optimized to maximize hydrolysis of cellulose and to maximize the yield of bioethanol. Hence, our objectives are to determine the optimal conditions for acid pretreatment and to obtain ethanol from cowgrass after acid pretreatment, cellulase hydrolysis, and fermentation. We hypothesize that pretreating cowgrass for 90 minutes with 2% sulfuric acid will have the highest glucose yield, and the ethanol can be obtained after acid pretreatment by cellulase hydrolysis and fermentation. Our independent variables are the concentration of sulfuric acid used to pretreat the cowgrass and the time taken to pretreat the sample, while the dependent variables are the glucose concentration. And here is the list of our control variables. Here is also a list of our apparatus used, and here is a list of our materials.
To carry out the experiment, cowgrass was added to sulfuric acid of varying concentrations. The mixture was boiled on a hot plate for different durations. It was left to cool and then centrifuged. The supernatant was then neutralized with 30% sodium hydroxide and analyzed for glucose using the DNS total glucose assay. The solid fraction was washed and dried to be used for further enzymatic hydrolysis. The solid fraction samples were hydrolyzed using cellulase. Sodium citrate buffer was also added to maintain the pH at 4.8. The samples were placed in a water bath shaker for 2 days. After filtration, the filtrate was tested for glucose using the DNS total glucose assay. It is then fermented with yeast for 2 days. Distillation was carried out to obtain ethanol, and the ethanol concentration was determined with an ethanol sensor. A control was also set up in which cowgrass was hydrolyzed without acid pretreatment. At the final stage of our project, cowgrass was pretreated with the optimum conditions and the supernatant was fermented to yield ethanol. The solid fraction was hydrolyzed and fermented to produce ethanol. Thus, we can determine the total ethanol yield.
For our results, we found out that for all durations, the increase in the concentrations of the acid increases the glucose yield. This shows the hemicellulose degradation after acid pretreatment. Glucose yields also increase when samples are pretreated for longer. However, this is not the case for 2% sulfuric acid, as there is a decrease in the glucose yield after 60 minutes. The prolonged pretreatment and the high concentration of the acid could have caused the glucose to be degraded, hence leading to a lower yield. The optimal condition of pretreatment is determined to be 2% sulfuric acid for 60 minutes, since it resulted in the highest glucose yield. Glucose was also released after cellulase hydrolysis, reflecting the digestibility of the pretreated cowgrass. The solid fraction obtained by pretreating the cowgrass with 2% acid for 60 minutes also gave the highest glucose yield. Therefore, this is indeed the optimum condition. The results from our control also showed that cowgrass that had undergone pretreatment before hydrolysis had a better yield than the control, which had not undergone any pretreatment. This shows that acid pretreatment is indeed effective in increasing the glucose yield. Under the optimal conditions, we obtained 1.78% of ethanol from acid pretreatment and 0.2% from cellulase hydrolysis. 3.97 milliliters of ethanol was obtained for every 100 grams of cowgrass. Our first hypothesis is rejected because pretreating cowgrass with 2% sulfuric acid for 90 minutes did not result in the highest yield. However, our second hypothesis is accepted and validated, as we have indeed successfully obtained ethanol.